So now that we've considered cell division in eukaryotic cells, let's talk about what happens when the control mechanisms break, and that is cancer, right? Now, cancer results from genetic changes that affect cell cycle control, right? Go back to the genes, which make the proteins. We know that these networks of proteins that control cell division and information from outside the cell are very, very complex. And that's important because that gives us lots of control. But it also then is lots of points that can break. So there are redundancies involved in that system, but at the same time, the redundancies can be overcome with lots and lots of little errors. Now, cancer basically are cells that are growing out of control, not unlike the cell growth that you see during rapid development, right? So we're going to be piggybacking on systems that are already exist, but are just turned on at the wrong time. So we lose control of the cell cycle. That is cancer. So cells may be making their own growth factor instead of having to receive it with a receptor on the outside of the cell. They may have a receptor that's broken, so it seems to them that the growth factor is always present. They may have other factors that are involved in the cyclin cell cycle control system that are broken, or other systems that are involved in cell cycle control. So all these proteins, any of them can be broken that contribute to a cell becoming cancerous. Now, there's some vocabulary that we need to introduce, and that is the idea of oncogenes and proto-oncogenes. So an oncologist is a cancer doctor. So onco deals with cancer. Oncogenes are cancer-causing genes. Now, proto-oncogenes are genes that are normal in the cell, but then when they're mutated, they become oncogenes. So these are the corresponding normal cellular genes responsible for normal cell growth and division, and when you break them, they become oncogenes. So the conversion of a proto-oncogene to an oncogene can lead to a breaking of the cell cycle. So how can that happen? So when we look at this, we can say, oh, look, you've got your regular proto-oncogene. Maybe you get a translocation event, right? So this can happen with viruses. You move genes around, and now you're under a new promoter, which is on more than it's supposed to be. You get an excess of growth-stimulating proteins. You may get copies of the gene form. Now you get an excess of growth-stimulating proteins. You may get control elements that come into play when they're not supposed to. You may get a different version of a gene that turns on the cell cycle when it's not supposed to. So these are all now numerous mutations, any of which can result in the same disease, which we call cancer. So again, movement of the DNA within the genome, amplification of the proto-oncogene, point mutation. So all the stuff we talked about that deals with gene control, expression of a gene, any one of those steps can go wrong with a proto-oncogene, and all of a sudden now you've got an oncogene. Now, the other thing is that not all of the genes that can break are involved in promoting cell growth. So some of them may be involved in telling the cell not to divide, right? So tumor-suppressor genes are genes that say, hey, we're not supposed to be dividing now for whatever reason. We'll talk about that. Many of them are they repair damaged DNA, they control cell adhesion, and they're going to inhibit the cell cycle in the signaling pathway. They're going to say, hey, I'm not getting all the stuff that we're supposed to get. My input is negative. Don't go forward. But again, as we know, because we studied signal transduction, you're getting input from many, many pathways in many cases, and it's going to be the summation of that signal that really dictates the red light or green light. Now, at the same time, we want to consider two examples of proteins that are key to controlling cell proliferation that you'll maybe hear about as you go on in your studies, and those are RAS and P53. So the RAS gene is a proto-oncogene that when it breaks, it can oftentimes lead to a hyperactive protein. So if we look at the G protein RAS, if it has a mutation, it's going to be on all the time. So when it's on all the time, it's sending a signal down the signal transduction cascade to say, hey, DNA, you need to turn on these genes that make proteins that stimulate the cell cycle. So normally it would need the input from the growth factor and its receptor, its corresponding receptor. But if it's broken, if it's mutated, it's like, hey, I'm on all the time. We're going. That's bad, All right. Now, alternatively, it could be a breaking protein that's broken, and so it's not able to suppress the cell cycle. So p53 is an example of that. In the case of p53, this is a protein that is turned on when the cell experiences DNA damage. So you don't want the cell to divide, because if the DNA is damaged, it may be damaging proto-oncogenes to make it cancerous. It may be damaged in other ways that you don't want that cell to go on and make copies of itself, because it's not going to be right. So normally this protein turns on and makes proteins that inhibit the cell cycle. If p53 is mutated, it may not work at all. So now all of a sudden the damage in the DNA may actually be in proto-oncogenes converting them to oncogenes, which normally then would be shut down, but in this case it's not, because you already have this other mutation that was unchecked. So when you look at this, increased cell division is kind of coming from both directions. You can kind of push the gas pedal and you get mutations that overstimulate the cell cycle, or you can take your foot off the brake and you get rid of proteins that are actively trying to put the brakes on the cell cycle. Both of these together are certainly going to increase cell division, and that then is going to be a big problem. Now, so this idea then is that you oftentimes need multiple mutations. There's an accumulation of mutations to ultimately develop in a full-blown cancer. This is one of the reasons why we don't often see cancer in younger organisms. It accumulates over life as you accumulate more mutations, ultimately getting the right combination that take the control off the cell cycle. All right. Again, going back to kind of the complexity of those signal transduction pathways, in this case the signal transduction from epidermal growth factor receptor, any one of these things could break and ultimately lead to mistaken gene transcription, proliferation, invasion, metastasis, resistance to apoptosis, programmed cell death. We want that to happen in some cases, and it's not. Angiogenesis, right? So all these things are bad. Okay. So a couple more words to add to our vocabulary list with regard to cancer. Transformation is the conversion of a regular cell into a cancerous cell. And a tumor then is a mass of these abnormal cells within otherwise normal tissue. Now, that happens, right? And it can be a benign tumor. A benign tumor then can be removed because all the cells that have the mutant DNA that are growing uncontrollably can be taken out. It becomes very, very difficult when a tumor becomes malignant and metastasizes. That's when the signals in the cell actually, for its own self-preservation, as it reproduces, invade surrounding tissue. Now, these can then break off and spread to other parts of the body and form secondary tumors. Right? So breast cancer here is an example where you have a tumor growing and then it metastasizes and spreads and becomes brain cancer or lung cancer or bone cancer or liver cancer, what have you. Right? So the thing about cancer is that it's a very, very complex disease, and it can be caused by many, many different sources. Now, people talk about inheriting a predisposition to cancer, and we have many genes that we're aware of, many mutations that we're aware of, that do increase your likelihood of getting cancer, but nothing that guarantees it. Right? So BRCA1 and 2 are genes that are well known to be associated with breast cancers. There are other genes, colorectal cancer genes, other genes that I can look at your genome and say, oh wow, you have a couple of these mutations that indicate you're already kind of on the road to accumulating the right number of mutations to result in cancer. Does that mean you're going to get it? Absolutely not. If you don't have these indicators, does that mean you're free from cancer? Absolutely not. It's a very, very difficult disease. Now, the other thing that makes cancer cells really, really interesting and kind of a conundrum is that their genome is all messed up. So if you look at this karyotype of a normal cell, a normal human cell versus a tumor cell, you can see numerous duplications, deletions, all kinds of recombinations and translocations of pieces of chromosomes. It is messed up. This is a cell that has one objective, and that is to reproduce itself. And that is cancer, and that's why cancer is so very, very difficult to combat.